Hello, I'm João Carias, and I'll be presenting evaluating rotation invariant strategies for mitosis detection through YOLO algorithms. A quick overview of the content of this presentation. First, we'll start with the motivation for this paper and the contributions of this work. Then, we'll go over the materials and methods where the main experience is detailed. Next, a section with the results from the experiment and its interpretation. Finally, we'll talk about the conclusions from this work. Mitosis counting is a useful tool for cancer detection as it is a good indication of its presence and aggressiveness. This task requires the time and attention of a specialist, which makes this an expensive procedure. Obviously, an automatic detection system is a natural fit for this problem. Presently, there are several object detection algorithms that are capable of detecting a given number of objects in an image, requiring a large number of examples of the same type of objects in other images. Specifically, our use case requires a large number of annotated mitosis examples for the system to have enough accuracy for it to be usable. Moreover, mitosis can appear in 4 different stages, where the last stage, telophase, is quite different from the others. An inexpensive way of obtaining more data is by augmenting the dataset. There are several types of augmentation, but in this work we focus on the rotation augmentation. For pathologists, the rotation of an image does not affect the classification of whether a structure is a mitosis or not, but for an object detection algorithm, it can be problematic. On the right, we can see an example of this. When the image is rotated, the algorithm shows inconsistent detections. The algorithm only learns from the image without rotations. So our objective is to obtain an object detection algorithm that is rotation invariant. The main contributions of this work are a rotation invariant strategy for the augmentation of a dataset and a comparison of YOLO 4 and YOLO 5 for the use case of mitosis detection. We chose 2 versions of the YOLO algorithm due to their known accuracy on detecting a large number of different objects, due to their open source code being actively supported, and due to their popularity. We created a dataset annotated at tau patch level with dimension 640 by 640 pixels. For this, we adjusted 2 datasets of breast cancer histological images, Mitosis atypia 14 and 2PAC16, for the object detection task. The initial dataset contained 2,502 images. We split in the following proportions: 70% training, 15% validation, and 15% test. Image on the right are dataset samples of breast cancer histological images. Mitosis atypia 14 on the top and 2PAC16 on the bottom. The green rectangles are the annotated mitoses. The rotation augmentation, while simple in itself, introduces some challenges. For the image rotation, for its size to remain the same, some part of the image has to be clipped. This means some mitotic events may be clipped or completely lost. To keep all the identified mitoses and avoid its duplication, for each image and corresponding annotations, we develop a simple procedure. Usually, this entails just rotating the image and annotations. But here's an example of the most complex case. When rotating the image on the left, and depending on the angle, mitosis near the border will be clipped or disappear. To solve this, instead of 1, 2 rotated images are created. The image in the middle contains 2 annotated mitoses. The image on the right contains a single annotation, and the area of the duplicated annotation is deleted. 3 strategies for the rotation of annotated bounding boxes were considered. In the first strategy, the new bounding box is calculated by finding the minimum and maximum xy coordinates of the corners of the rotating bounding box. In the second one, only the center point of each side is considered, while in the third, each side is divided by 3 and only the midsection is considered for the new bounding box. For the first strategy, the area of the bounding box in some cases increased significantly. For the second one, the inverse was observed, often clipping relevant areas of the target object. A reasonable middle ground was found in the third strategy, keeping the relevant parts of the mitosis inside the new bounding box while minimizing the increase of its area size. In this work, we used the third strategy. This table shows the datasets that were created and used in the experiment. The base dataset was not augmented. It contains 1,751 training images. The next dataset has a rotation augmentation with a 22.5 degree angle, greatly increasing the training subset. To the flip/4 dataset was applied a horizontal flip augmentation and 4 rotations multiples of a 90 degree angle. The flip/36 dataset was augmented with a flip and rotated by multiples of 10 degree angles, containing a training subset 72 times larger than the training subset of the base dataset. Finally, the test subset was transformed according with the flip/360 parameter, flip and exhaustive rotations of 1 degree angle, occupying around 170 gigabytes. The table shows the different models that were trained and tested. Both versions 4 and 5 of YOLO were trained in the previous dataset combinations. From the base dataset, 2 models for each version were obtained, a model trained without augmentations and another trained with online flip and online rotation. A similar strategy was applied to the 16 rods dataset. Models were trained with and without online augmentations. At the startup of the training, we applied transfer learning using pre-trained models, YOLO v4 custom and YOLO v5x. Also, we implemented online rotation augmentation in YOLO 4 to guarantee equal conditions in the comparison with YOLO 5. Here we can see the main results. Each one is the average of 3 runs. The baseline models trained with the base dataset show a F1 score of over 80%. Just using online flip and online rotation augmentation with this dataset improved the result by 4% in the case of YOLO 4 and 6% in the case of YOLO 5. Using the dataset of 36 rotations shows an improvement comparing with previous models. Similar results were obtained with the dataset with 16 rotations after applying online flip augmentation. Moreover, adding a small interval of online rotation augmentation decreased the F1 score of YOLO 4 but improved on YOLO 5. Generally, the version 5 shows an improvement over version 4 by 2%. When online rotation augmentation were used, the improvements of YOLO 5 rose between 3 and 4 over YOLO 4. Finally, YOLO 5 takes longer to train. To better analyze the results, we focus only in the FUM score, which is arguably the most useful metric for our use case. Each rectangle under FUM score is a comparison between YOLO 4 and YOLO 5 with the same parameters. The best result, shown in green, is consistently obtained by YOLO 5. The benefits of our proposed strategy can be seen by comparing the second set of results with the third. There's a relevant increase in performance for both V4 and V5 models, which indicate the strategy usefulness. Notwithstanding, this is at the cost of a significant decrease in dataset size, since it contains 36 rotations for each original image. Interestingly, removing the offline flip augmentation and decreasing the number of rotations from 36 to 16 did not decrease the FUM score in both models, while taking the same or less time to train. When using online rotation augmentation, the different architectures have different behaviors. Our implementation of online rotation augmentations in YOLO 4 improved significantly the performance when used on the base dataset, more than 3%. But when used with a dataset of 16 rotations, the performance actually decreased. This is shown in the last result. For YOLO 5, both cases had a better performance when using online augmentations. Here we have one example of a slide patch where some mitoses are present. The green rectangles are the detections of the model. On the left, a YOLO 5 model was trained with the baseline dataset. Without data augmentations, the model is not able to detect the same mitosis for all rotations. On the right, the best obtained model, a YOLO 5 trained with a mix of online and offline rotation augmentations. Its detections in the example are very stable. This example is illustrative of the obtained results and the importance of this work. The proposed strategy was a step forward in the objective of rotation invariance for the use case. From this work, we conclude that the proposed rotation augmentation procedure improved the performance of the tested object detection algorithms for mitosis detection. A mix of online and offline augmentations helped YOLO 5 reach the best observed performance. Additionally, this architecture showed consistently better results than YOLO 4. Finally, here are the references for YOLO 4 and YOLO 5, and for the used datasets, Mitosis Atypia 14 and 2PAC 16. Thank you very much for your time and attention.